Ultrafiltration is the filtration of the blood at a very high pressure, tremendous pressure, extraordinary pressure. So now, this is the basic way of writing. What is ultrafiltration? Filtration of the blood under tremendous pressure. Now, which part of the nephron are we talking about? Where does the process of ultrafiltration takes place? The process of ultrafiltration takes place in the Bowman's capsule and the glomerulus, or I would say the entire process of ultrafiltration majorly takes place in the glomerulus. We had defined the word glomerulus earlier. What is the definition of the word glomerulus? Glomerular capillaries are a network of blood capillaries. Here, I am drawing only one such capillary. Only one such capillary I am drawing. But how did we define earlier? A glomerular capillary, what is the definition of the glomerular capillary? Formed by the division of the afferent arteriole. What are glomerular capillaries? Define glomerulus. What will you write? Define glomerulus. A network of blood capillary, a knot of blood capillary formed by the division of the afferent arteriole. Okay. Now let us try understanding what is ultrafiltration and why the high pressure. First of all, the blood is flowing towards the nephron, so definitely it is brought from an artery. So before the afferent arteriole, if I have to draw one more blood vessel, then which blood vessel am I supposed to draw? If this is the afferent arteriole, try to recollect what we had studied the last day. This is the afferent arteriole. And before the afferent arteriole, if I have to draw one more vessel, for example, if I have to draw one more vessel here, which is this vessel that I want to draw here? Which is this vessel? The renal artery. So you are already aware that when we are talking about the renal artery, or if it is the afferent arteriole also, the pressure of the blood in an artery is always very high. So this is one reason. Second is, this afferent arteriole and the efferent arteriole, they don't have the same diameter. There's a huge difference in their diameter. The afferent arteriole is very wide. The afferent arteriole is wider. And the efferent arteriole is much narrower. So since this is a wide tube bringing a lot of blood into the capillary network, but that same amount of blood cannot go out. So definitely there is a surplus amount of blood here always, and now again adding of blood and again adding of blood. So this is what is resulting in the development of a pressure. Development of a pressure, that is also called the osmotic pressure. So now what is happening is, what are the reasons for the development of pressure, high pressure in the glomerular capillaries? What is the reason? Development of high pressure in the glomerular capillaries, the uneven diameter of the afferent and the efferent arteriole. That is one of the clearest and the specific answer that you should always write. What is the most important reason? There are few more reasons, but for the 10th standard, you are writing this point, that is more than enough. What is the most important reason for the high pressure of blood in the glomerular capillaries? Uneven diameter of the afferent and the efferent. Meaning what? The afferent arteriole is very wide, broad, bringing a huge amount of blood every time, but that much of the blood cannot be drawn out from the nephron by the efferent arteriole because it is narrower. So just you can imagine of a place, for example, now that the entry is wide, big, and the exit is narrow, what is happening? There's a crowd getting formed. No. So if you think of your school gate, at one particular time, maybe 10-15 of you can enter, but then exit gate is so narrow, only 3 people can go out. So definitely a crowd. So something like this, you think that the entry is quite broad, the exit is very, very narrow. Now that is the first thing we are understanding. Ultrafiltration, why the high pressure? High pressure is because of the uneven diameter of the afferent and the efferent arteriole. But after that, now that we are talking about that the uneven diameter of the afferent and the efferent arteriole is one big reason, Why? There is a huge high pressure in the glomerular capillaries. After that, what? The glomerular capillary, we are talking about capillaries. If you would have done circulatory system earlier, then by now you are already aware that if it is a capillary wall will always be very thin. If it is a capillary, any part of the body, if it is a capillary, the capillary will always have pores. So now, when we are talking about the glomerular capillaries, every particular capillary wall has pores. Every capillary wall has pores. Now that I talk about, and the capillary wall is always very thin. So now that the capillary wall is thin and now that the capillary wall has pores, now what is happening? The liquid part of the blood is oozing out. The liquid part of the blood is oozing out. The liquid part of the blood is coming out from here. And this liquid part of the blood is all getting collected here in the space of the Bowman's capsule. The Bowman's capsule is just like a cup-shaped structure. The Bowman's capsule is like a cup-shaped structure. It has got a cavity there. So the blood is rushing through these capillaries. As the blood is rushing through these capillaries, what is happening? As the blood rushes through these capillaries, since the capillary wall is thin, quite a number of salt ions can diffuse. Since the capillary wall has pores, the liquid part of the blood can come out. What is the liquid part of the blood known as? The plasma. The liquid part of the blood that I am mentioning, that the liquid part of the blood is all coming out and gathering where? Coming out and gathering in the inner cup-shaped depression of the Bowman's capsule. Then, what is this process known as and what is this fluid known as now? What is this process known as which gives rise to this fluid? The process is ultrafiltration. And what is this fluid now known as? This fluid is now known as the glomerular filtrate. The fluid is known as glomerular filtrate. What is the composition of this glomerular filtrate? Very important to know. The composition of the glomerular filtrate is a huge amount of water, large amount of salts, large amount of glucose, and if I talk about urea and uric acid, etc. are in traces. So what is the basic composition? I'm saying what is this fluid known as after the process of ultrafiltration, the fluid that is formed, what will be termed that fluid as, which is getting collected in the cup-shaped depression of the Bowman's capsule. The fluid is known as the glomerular filtrate. What is its composition? It has got a huge amount of water. Salts like sodium, chloride, potassium, etc. and glucose, huge amount of glucose. Sodium salts majorly. Sodium chlorides majorly. Urea, uric acid, is it a component of the glomerular filtrate? Yes, but in traces. So now think, if this blood which was coming or entering the nephron has already given away almost about 90% of its water, then the blood which is entering or going into the efferent arteriole, is the blood comparatively thicker? Is this blood here in this vessel comparatively thicker? Is the blood in the efferent arteriole comparatively thicker? Yes. The blood in the efferent arteriole is comparatively thicker. And what is its composition? What is present in the blood of this capillary? Which capillary am I talking about this? The entire surrounding network of capillaries. What is it also known as? Also known as, this is one I have written. What is it also known as? The vasa recta. So the secondary network of capillaries or the vasa recta, then what will be the composition of the blood here? Number one, the blood has very less amount of water because ultrafiltration. As a result of this process, so much of the water is given away. So what is less and what is more here? The water component, if I would say that the water component in this blood is very, very less. What about the water here? The water is very less here now present. What is present in large quantities? Present here in large quantities are urea and uric acid. What is present in large quantities? Urea and uric acid in large quantities present. What about this glomerular filtrate? The other day already I told you this entire tubule, that is the Bowman's capsule and the tubular part, if you understand, the inside is all hollow, just like the intestinal tubes. The inside is all hollow. So what will happen to this fluid? The fluid is all entering inside the hollow of the tube. This entire tubular part, now for understanding, I would divide into three parts. This is the PCT. What is the full form? Proximal convoluted tubule. What is LH? Now that I write, what is its full form? Loop of Hadley. And the last part is the DCT, that is the distal convoluted tubule. So this entire region I divide into three major parts: the PCT, the loop of Hadley, the DCT. Try to understand what is happening with the components here and in the blood. So now the glomerular filtrate which is running down here, what does it have in large amount? It has got a huge amount of water. It has got a huge amount of water. The water content is so much here in the tubule, less in the blood. What about the sodium ion? Sodium ion so much here. Sodium is so much here. What about the glucose? Glucose is also so much here. So now the glucose and the sodium, these components are so much here in the entire tubular region. In the tubule and in the blood, we are just comparing, and this is what is actually happening in our nephrons. Surrounding the nephron, there is a huge network of blood capillary. The wall of the capillary is thin. The wall of these tubules also, they are permeable to different type of ions and components. What is the meaning of the word permeable? Allow the transport possible. Transport. Allow transport possible. So I am saying the PCT, the loop of Hadley and the DCT have differential permeability. And this entire capillary network, they have fine thin walls which allow the process of diffusion possible. Because of that, the exchange of substances now go on. So for ultrafiltration, what did we see? And after ultrafiltration, what is the product? Glomerular filtrate. How much of the glomerular filtrate formed per day? 160 to 180 liters per day. 160 to 180 liters of glomerular filtrate per day. How much of the blood do we have in our body? 4.2. 6 liters at the most. 5 to 6 liters. But how come we are talking about 180, 160 liters of the filtrate per day? Because almost 300, 400 times the blood constantly gets filtered. I may talk about 100 ml of blood. Is there 100 ml of blood? Is it getting filtered only once in a day? No, almost 400 times. In a 24-hour span, almost 400 times that one single small amount of the blood gets filtered. So that way it is counted that almost about this much, 160 to 180 liters of glomerular filtrate. Now, so we are understanding that this 180 liters of filtrate, if it is, can our body afford to lose away this much of the liquid? No. If that is so, then after every time when the body is releasing out the waste substances, the urine, then we would suffer from massive dehydration. We would suffer from massive dehydration, but generally does not happen because now that the glomerular filtrate is in the tubule, the surrounding blood and the composition in the surrounding blood capillary is absolutely disturbed. Can this happen? Can this remain for a long time? No, within few minutes it has to be restored. Within few minutes the composition of the blood has to be restored. What am I saying? Here now the blood has a whole lot of urea, uric acid, water very less, glucose very less, some amount of proteins there. So the composition of the blood is not regular. It has to be restored back. So what will happen immediately? immediately a process known as diffusion happens, where a whole lot of sodium starts moving, diffusing from the PCT towards the blood capillary. A whole lot of water starts diffusing from the PCT towards the blood capillary. Some amount of glucose starts diffusing from the PCT towards the blood capillary. What is this process I mentioned? Simple diffusion. What is the rule for diffusion? What is the rule for diffusion? Molecules will always move from a region of high concentration to a region of low concentration. So that is what is happening here. The tubule has got so much of water, so much of glucose, so much of sodium. It is the glomerular filtrate only which is coming down. Since its composition keeps on changing now, after some time this is no more known as a glomerular filtrate actually. Now it is also known as the tubular filtrate, because the moment the process of exchange starts, the composition here and the composition here is different, right? So now it is no more known as the glomerular filtrate, rather from the PCT it is known as the tubular filtrate. So what did I tell you? What is happening majorly in the PCT? What is happening between the PCT and the nearby blood capillary? The diffusion of sodium and the glucose and huge amount of water. So what is this second step known as? Second process known as? The second process is known as the selective reabsorption. The second process is known as selective reabsorption. So what is happening in selective reabsorption? Molecules are moving. The molecules are moving from the tubular part of the nephron. Molecules are moving from the tubular part of the nephron. From the tubular part of the nephron, where are they going? They are going to the capillary network, the capillary blood. What is this process happening? The process is nothing but diffusion. So now I say that what is selective reabsorption actually? Selective reabsorption is the movement of the essential ions and a huge amount of water from the tubular part of the nephron towards the blood. How is it possible? Because this PCT membrane is extremely permeable to water, sodium, and this blood capillary network or this blood capillary wall, every wall of this blood capillary network that we know is extremely thin, so diffusion is possible. So that is the reason the water and all this. So now progressively what is happening? The amount of the water in the blood is getting restored. The blood is restoring back its water content slowly. Small amount of glucose coming here, a whole amount of the sodium ions now getting its entry into the blood. So now that we talk about this entire process, a particular hormone is responsible for this. What is this hormone known as? This is known as the antidiuretic hormone. This antidiuretic hormone is released from a specific organ, a specific gland, and this is known as the pituitary gland. We will talk about this later. But as of now, when we are talking about that the pituitary gland is one such endocrine gland in our body, and this endocrine gland is constantly releasing a specific hormone. What is this hormone known as? ADH. The full form, again in endocrine glands, in that chapter you are going to come across. ADH, also known as antidiuretic hormone, is majorly responsible for this process. Is this process necessary? Yes, because within a few minutes, the volume and the composition of the blood has to be restored, otherwise we will start suffering from some abnormal conditions of the blood supply, organ functioning, etc. So the moment ultrafiltration has happened, the hormone starts getting released and this will continuously happen. Reabsorption continuously happen. What is the concept of reabsorption? I told you, essential substances diffusing from the PCT towards the blood capillary. Then what in the loop of family? So by the time the fluid has come down to the loop of family, the fluid is still having a huge amount of water, and the blood also has not restored back all its amount of water and salts. So at any time, whenever we are comparing the loop of Henle and the surrounding blood vessel, what is the major process happening in the loop of Henle? The major process which is happening in the loop of Henle is just this, that is reabsorption of the water. So the entire concept of reabsorption majorly happens in two parts of the nephron. Which are the two major parts of the nephron? The PCT and the loop of Henle. Which hormone regulates this entire process? ADH. So I just now mentioned that ADH is regulating the process of reabsorption. Then what would happen? If a person has a deficiency of ADH, then what would happen? If I come across that if the secretion of ADH is very less, then what would happen? The amount of fluid which is there in the tubular part of the nephron, will it go back to the blood or it will get all excreted out? What did I tell you just now? That ADH is responsible for the reabsorption. This movement towards the blood, this is what is called reabsorption. I asked you that if the ADH secretion is less in an individual, then will this reabsorption happen efficiently or this entire amount of the water will be excreted out? Excreted out. So the person now starts suffering from a condition known as diuresis. Diuresis. When a person has a deficiency of ADH, what is ADH meant for? Reabsorption of the water and the sodium and the glucose back to the blood in a very short period of time, so that the blood volume and the concentration is maintained. Even after the process of excretion, the blood volume and concentration should be maintained within few minutes, then there is nothing to be worried about. So now that if an individual has a low secretion, maybe there is a problem with the pituitary gland, we come across this later, that if there is a problem in the pituitary gland, the secretion of the hormone is insufficient, deficient, then what will happen? The person will suffer from frequent urination, a condition known as diuresis. You have to remember this word, diuresis. Okay, now so we had just now talked about the concept of reabsorption. What did I say? What did you understand by the concept of reabsorption? Reabsorption means a whole lot of movement or diffusion of water and sodium and glucose from where to where? Tubule to the blood. Tubule, which parts of the tubule? The PCT and the loop of Henle, and going to the blood. But we were also saying that the blood here has high concentration of urea. Yes or no? I mentioned this also, that the blood here in the secondary capillary network, the blood here, the blood capillary surrounding the PCT, surrounding the loop of Henle, has a high concentration of urea. But I am not explaining you this, that the urea moves from here to here. Did I say that? Repeatedly I am saying that it is diffusion and may follow the rule of diffusion where the water moves from here to here, high to low concentration. Sodium moves here to here, high to low concentration. Then the rule for urea, uric acid should also be true, high to low. But no, I cannot say that the urea and the uric acid moves from here to here if it is the PCT, here to here if it is the LH, because the membranes of the PCT and the LH are impermeable to urea, uric acid. Never mind, here the concentration is high, but can they move? No, can this urea and uric acid move this way? No, they cannot. They cannot. Why they cannot? Because the membranes of the urea and the the membranes of the PCT and the LH, they are not ready to pick up the urea, uric acid molecules from the blood. What is that property called? Impermeable. They cannot allow, or they do not allow the movement of the urea, uric acid into the tubule. So this process does not happen. So back here now, when I compare the DCT and the surrounding blood capillary, that's the last part we are left with. Now with the DCT and the surrounding blood capillary, what happens? In the DCT and the surrounding blood capillary, we understand, yes, still there is a huge amount of urea and then uric acid and then potassium ions and then some poisonous chemical substances which enter our blood along with medicines.
The antidiuretic hormone maintains the difference level, or what does it do?
Antidiuretic hormone will make the membranes permeable so that the sodium and the water and the glucose can constantly keep on moving towards the blood. Understood? It actually brings about the diffusion possible. So now that I talk about the DCT and the surrounding blood capillary, now what the situation is, now here is the water content quite a lot. Now the water content is comparatively lesser. What about the sodium and the glucose, which are also not waste substances? What about the sodium and the glucose? The sodium and the glucose almost in traces now. So I am now talking about the DCT and the surrounding blood capillary, and this is the situation more or less. The blood has a huge amount of urea, uric acid, potassium ions, soluble vitamins, all the poisonous chemical substances that enter our body along with food, medicine, antibiotics, etc., all this come under this category. So now these components are more in the blood, less in the tubule. So what should the process be? Here the movement is in this direction. The urea comes here, the uric acid comes here, all the antibiotics and etc., they will be releasing into the tubular region. So what is the story here? The blood capillaries starts. Now which blood capillary I am talking about? The blood capillary surrounding the DCT. When I compare the DCT and the surrounding blood capillary, the urea and the uric acid moves towards the DCT from the blood. The urea uric acid is moving from the blood capillary towards the DCT. The antibiotics and all the poisonous chemical substances which are left in the blood starts moving towards the DCT. The soluble potassium ions and extra vitamins which are there in the blood starts moving towards the DCT. So ultimately now in the DCT, what is the situation? If I talk about them, in the DCT, what is the third step that is happening in the DCT? The third step, this is known as tubular secretion. What is the definition of tubular secretion? Then the definition of tubular secretion is diffusion of harmful toxic substances from the blood capillary towards the DCT. This is tubular secretion. So then in the DCT, if I have to now continue with the DCT, then what is the composition of the fluid now in the DCT? The composition of the fluid in the DCT is, it has a small amount of water. It has a large concentration of urea and uric acid and potassium ions and harmful drugs and poison. What about the sodium and the glucose? Is it there? Just in traces. Just in traces. And water, only that much whatever is required to dissolve all these components. And what is this fluid now known as? Can we still continue to term this fluid as the tubular filtrate that we were talking about? No. Now this fluid is known as the forming urine. This is known as the forming urine. Which tubule am I talking about this as? This is the DCT. I am talking about this tubule, the continuation of this tubule there. This is the DCT, and from the DCT, where will it go now? From the DCT, it will go to the collecting duct. From the collecting duct, where will it go? From the collecting duct, it will go to the pelvis. It will go to the renal pelvis. From the renal pelvis, where will it go? Ureter. From the ureter, where will it go? Urethra, and out from the body. So we were talking about this particular fluid now, which is there in the last part of the tubule, that is in the DCT, where substances started moving from the blood towards the tubule, and if this is the process, this is what is tubular secretion. If substances are moving from The tubule towards blood, that is reabsorption. And when substances are moving from the blood towards the tubule, this is secretion. And now the composition of this tubular filtrate is massively changed. So what is the composition roughly? It will have an adequate amount of water, what is just necessary to dissolve all the components, and the urea, uric acid, the potassium ions, glucose and sodium just in traces, and proteins in traces. So that is the composition that is getting formed now where in the DCT, which is now getting conveyed to the collecting duct. So this is what we are talking about as the forming urine, and in the collecting duct it is the urine now. The fluid that is flowing in the collecting duct almost to be conveyed to the renal pelvis, this is the urine. Now we are talking about this particular fluid formed by the filtration of the blood, How much of the urine per day normally to be released out? At the most 1.5 to 2 liters per day, not more than that under normal conditions. And we were talking about 160 to 180 liters of the glomerular filtrate. So can you understand 160 to 180 liters and now 1.5 liter, almost 98% of reabsorption we are talking about. Which is this hormone which is responsible for this, the ADH, which plays a very, very important role in reabsorption.
What happens when the ADH is secreted in excess?
What happens if the ADH is secreted in excess? What will happen? The reabsorption is even more faster. If the reabsorption is even more faster, quite a number of times it is happening, but that is comparatively rarer condition. Yes, in that process, what will happen? The urine flow of that person will be much lesser. And that person still will be having a tendency urge of drinking more water. So, how much of the urine per day normally? 1.5 to 2 liters. What is the color of the urine? The color of the urine is slightly yellowish because of a pigment known as urochrome. This word you will have to remember. What is this urochrome? Urochrome is a pigment formed by the breakdown of the bile pigments in the liver. The bile pigments, in the concept of digestion in the 9th standard, we have studied a little bit there. The bile pigments, which are the bile pigments? Bilirubin, biliverdin, they again get broken down, resulting in the formation of a simpler compound known as urochrome, which gives a yellow color to the urine, the slight yellow color to the urine. What about the pH of the urine? pH of the urine is basically 5 to 8, 7.58 at times, 5 to 7, 7.5 generally. So we would say that it is slightly acidic. The acidic, though sometimes we understand that it is acidic in nature, but that also very mild acidic feature that the urine has. And next is when we are talking about that, the volume, the color, the pH, and a specific order, because sometimes when we understand because of the intake of the protein, individuals where intake of the protein is more, the acidic nature may increase. That is again depending on the diet. This pH that I talk about is variable depending on the diet. If a person is extremely on a high protein diet, it will be, the pH will be lesser. The pH may be 5 to 6, 6.5, that also. So when we are talking about that, the pH would be lesser depending on the diet. And then apart from that, these are some properties that you have to remember. How much of the urine normally, what is urochrome, what is it responsible for, and the pH, slightly you have to remember this. And the order, the specific smell is because of some ammonium salts which are there. So now this entire process that we are talking about, what is this process we are learning? We are actually talking about the process of filtration of the blood. But along with filtration of the blood, what is happening? Just after filtration, ultrafiltration, what was happening? The composition of the blood was getting all disturbed. By this process of reabsorption and secretion, what is happening? The volume and the composition of the blood is getting restored back. So what is this process known as? This process is known as osmoregulation. What is osmoregulation? Is it a different process apart from urine formation and filtration of the blood? No, it is not a different process absolutely. The process of osmoregulation means the maintenance of the salt and the water balance. Maintenance of the salt and water balance in the blood. So how is this salt and the water balance maintained in the blood? It is because of the entire process of reabsorption majorly plays an important role. Reabsorption and tubular secretion is what is responsible for maintaining back the composition volume of the blood. So osmoregulation and filtration of the blood actually are parallel processes. It is not absolutely different a process. Since these processes are happening, so blood regains back its normal composition and volume. What should be the normal composition of blood? Adequate amount of glucose and sodium, proteins, an adequate amount of water. Blood should be 5 to 6.
Difference in diameter.
Structural difference when asked in the paper, structural difference only to write. Number one, I ask you write difference between afferent and efferent based on the structure, structural difference. Wider diameter, narrow diameter. Based on the type of the blood that they contain. This has got oxygenated blood rich in or high concentration of urea and uric acid. The blood is in its fluid state. What about this side? The blood is thicker. Amount of water is much lesser. And concentration of urea and uric acid is very high. Yes or no? So where will